Hello and welcome to the Talking Precision Medicine podcast. In this series, we sit down with experts on the application of AI and big data analytics in the drug discovery space. Our guests are innovators, business decision makers, and thought leaders at the intersection of data and therapeutics. We discuss the promise, practice, challenges, and myths of AI in precision medicine. This show is brought to you by Genialis, and Rafael, its CEO, is your host. Genialis is focused on data integration and predictive modeling of disease biology to help accelerate the discovery and de-risk the development of novel therapeutics. Today we speak with Patrick Hanley. Patrick works at the Children's National Hospital in Washington, D.C., where he serves as the Director of Good Manufacturing Practices for Immunotherapy. He is also a co-founder and vice president at Mana Therapeutics, a cell therapy company harnessing our immune cells to combat cancer. He is an expert in designing and manufacturing cell therapies and was once a fierce competitor on the Houston indoor soccer circuit. We talk about the leaps and bounds of progress in cell therapy and the challenges, including discovery, manufacturing, and distribution that lay ahead. Let's get right into it.
Hey everybody, today I'm here with Dr. Patrick Hanley. Patrick is the Director of Good Manufacturing Practices for Immunotherapy at Children's National and George Washington University. He's also a founder and vice president of a new and exciting cancer cell therapeutics company called Mana Therapeutics. Patrick, thanks for joining me.
Yeah, thanks, Raf.
So why don't you, let's start with either or. Let's start at Children's National and GW. Tell us a little bit about the center where you work and who all is there and what you guys are up to.
Yeah, sure. So we are a cellular therapeutics program that started in 2013 under the direction of Dr. Catherine Ballard. Three of us, or a number of us, but three faculty members moved from Baylor College of Medicine in 2013 to start this new exciting journey. And I'd say there were about maybe 12 of us when we first started, and now we're up to probably more than 40 people who all work on different cellular therapies for cancer and a number of various diseases.
Wow, that's a pretty big group now. And how is the group organized? So I introduced you as being the head of GMP, of manufacturing practices. Can you talk a little bit about the different kind of divisions and sort of how the labor works?
Yeah, sure. So obviously, like I said, Dr. Ballard is our programmer and center director. And then there's a couple of directors who are involved in different stages of development. So we have a translational research laboratory that's under the direction of Dr. Russell Cruz and Dr. Michael Keller. And they do very basic science discovery. So, hey, I found a new way to generate T cells that can target, pick your favorite virus or pick your favorite cancer. And what happens then is they work closely with my team and also the regulatory director, Famita Hawk. And we say, okay, we want to translate this to a clinical trial. And so here's what we need to do to do that. So Famita is intimately involved in the regulatory strategy for that. Here's what the FDA is going to require, that sort of thing. And our team says, okay, so you have this strategy for making the T cells that target, let's just use leukemia, for example. So you want to target leukemia with these T cells. We looked at your process. It looks like it's pretty straightforward, but we noticed that you use fetal bovine serum in your manufacturing. Well, there are certain regulations that say that you need to use US herds for that fetal bovine serum or from New Zealand. And so we do kind of that risk-based analysis to make sure that the way you're manufacturing is in compliance with good manufacturing practices, in particular for phase 1, because there is a sliding scale of GMP.
Huh, that's fascinating. Yeah, I was, reading the other day, and this is something that a lot of us who come either from the just laboratory research side or the data side don't think about. But so much of our kind of future of precision medicine is biologics, and biologics is a really big manufacturing challenge. It's almost a whole new set of things that a lot of us hadn't really thought about before.
Yeah, that's right. So I'm not sure if you're familiar with the new FDA chairman, Scott Gottlieb. He's actually come out and said, traditionally it's been 20% manufacturing for new drugs and 80% of the clinical protocol. Now with these new biologics, such as CAR T cells, chimeric antigen receptor T cells, 2 of which were approved last year, and they're the first cell and gene therapies approved ever by the FDA. But these products are now 80% manufacturing and only 20% of the clinical study. And I think that speaks well to what you're talking about here is, personalized medicine is just sort of turning traditional pharmaceuticals on its head.
Yeah, no, it's incredibly exciting. Let's talk a little bit about those FDA approvals. So I followed that as well. There were 2 CAR T therapies approved last year. Should we expect that to be sort of an opening of the floodgates and we'll see lots more of this? Or are we still at a point where these are sufficiently hard to develop and prove that it's going to kind of new therapies will trickle in for a while?
Yeah, so I think it's probably going to be both. I think there's going to be opening of the floodgates. There's going to be a number of new approved therapies in the next year or two, one of which will probably be from Juno. So it'll be the third CAR T cell that's targeting CD19. B-cell leukemia, it's an important disease that we need to find a way to cure. But let's be honest, there's still only a limited number of those patients that we can treat. So if we have 3 CAR T-cell products that are all treating the same diseases, that's not going to be beneficial for the system. I should point out that they are 3 of the same types of cells, but they probably will likely target different indications. So Kymriah, which is from Novartis, is targeting refractory and relapsed leukemia. Yescarta, which is from Kite Gilead, is targeting DLBCL type of lymphoma. Kymriah did get a secondary approval for DLBCL, but you can see where I'm going with this is, they're still targeting a very small subset of hematologic malignancies. And so, the holy grail is twofold for these CAR T cells anyway. Solid tumors, because there's a lot of different types of solid tumors. And the second is an allogeneic approach. So right now, this is truly personalized medicine. So every patient gets a different cell, and those cells come from their own body. But the allogeneic approach is let's make a bank of these cells, and then they'll be immediately available, just like aspirin, just like your chemotherapy. Those are already in the bank and ready to go.
Interesting. So basically turning these into almost what we think of more as drugs rather than something where a patient has to go in and have their own cells pulled out and engineered and so forth.
That's right. And I'm not saying that they have to be allogeneic. I think that there is utility in a lot of these modalities. But right now, these things are expensive, right? So Kymriah costs $475,000. Yescarta costs, I think, $373,000. And that's not cheap. Medicine is definitely getting more expensive. But no sort of groundbreaking transformative technology started out being incredibly cheap, right? Whether it be the Model T, as Bruce Levine likes to say, or your television and computers, all of these things were really prohibitively expensive. But now I think a lot of people can afford them.
Sure. No, that makes sense. We're very much in the early days. How do you think scientists in the industry think about developing sort of new cell therapeutics for diseases? Like, how are the indications chosen? Is it a question of simply where we have the most scientific maturity, or are there market considerations? I mean, I guess at SETI, how do you guys decide what to work on?
Yeah, so in some ways it's a me too. Novartis came out with their CD19 CAR, and Carl June is a brilliant guy, and in that group was very successful. But there were 5 or 6 other teams who were developing CAR T cells at the same time, and he picked the right costimulatory domain to make it work. And then there was a lot of money thrown at it, and then you see a lot of other companies spawning up who also want to target CD19 and other indications with the CARs. But in terms of how you choose, right? So I think also it's where is there an unmet need? So I should point out that I am employed by Children's Hospital, but we also have a startup, Mana Therapeutics, and so my conflict of interest management plan dictates that I need to let you know when I'm speaking on behalf of which entity. In terms of the CARs, it's a low-hanging fruit. So CD19, which is what the CAR T cells target, it's expressed on the cell surface of those B-cell malignancies. And so it's very easy for an antibody or a CAR T cell to recognize that tumor cell. And so that's why you see all these companies coming out with the CD19 CAR first, as they secretly try and work on identifying targets for solid tumors.
Interesting. Since you've already identified, we'll talk about Mana. We can stay here for a little bit, just so you don't have to keep switching back and forth. Tell me a little bit more about Mana. So this is Dr. Ballard and yourself, and who else is involved in this company, in this project?
Yeah, so Dr. Russell Cruz, he's the director of our preclinical lab that we talked about earlier. And then, we were really blessed to identify sort of business co-founder, Mark Cohen. He's on the board of Dana-Farber, and, he's sort of a serial entrepreneur, if you will. He has a number of successful startup companies. He's the one who really pointed us in the right direction and say, here are the right steps you need to take so that we can get this to more patients faster.
Yeah, no, Mark Cohen's kind of a lion in the industry. We actually work with one of his portfolio companies in particular and pay a lot of attention to the kinds of things he says and what he does. He seems to be a kingmaker in this space. That's neat. And so you guys have, I'm looking at your website now, this is Mana Therapeutics, and it looks like you have plans to move into, as you said, solid tumors, the holy grail, and some secondary indications.
Yeah, that's right. We also have T cells that are targeting a number of different viruses as well, or virus-associated malignancies. So, we're still sort of in stealth mode. So we're hoping to maintain that stealth for a little bit longer, but I think big things are in store.
Let me just ask a scientific question, and this exposes how long it's been since I was actually in a lab and kind of reading the literature. For hematological cancers, it's kind of obvious to me how you might deliver sort of a cellular therapy. Does delivery become harder for solid tumors, or is it just a matter of injecting to the right spot?
That's a good question. I think I'll answer it now with our current knowledge, but things could change, right? You know, if you're targeting a brain tumor, maybe it's necessary to utilize an Amaya or something like that so you can go directly into the brain. But right now, we're targeting solid tumors that seem to be accessible by intravenous infusion. One of the beauties of T cells is that they have receptors on their surface that allow them to home to sites of inflammation, which would be include your tumor. If you're using other molecules or even nanoparticles, they don't have that same homing. And so it's a lot more difficult. You have to give a more targeted approach to delivery.
So you can program the T cells to not only hopefully kill the tumor, but find it first.
Yeah, that's right. So, there's a lot of interesting studies in mice where you insert the tumor into one flank and you give the T cells IV or even into the other flank. And you can see that actually the T cells will then home to the tumor site on the other flank and still eradicate that tumor, oftentimes finding the additional metastases as well.
Oh, that's fascinating. So one of the aspects of cell therapy that's so exciting to me, and maybe this is, again, my incomplete understanding, but there's a promise anyway that once a patient has been treated with a cell that can recognize and kill their cancer, their body will continue to produce those cells and essentially cure the cancer. Is this true, or is this at least in principle the goal? How far are we scientifically from actually having a cellular cure for these tumors?
No, I think you're absolutely right. I think I hesitate to say that it's similar to vaccination because I think, there's a lot of unfounded stigma around vaccines, which again are not true. But really what we're doing is we're doing the vaccine in our laboratory and then giving it the end product to the patient. And so we do expect these cells will be around lifelong. We haven't been around long enough to know that for sure, but there are a lot of interesting studies with EBV specific T cells. And, EBV specific T cells are unique because not only do they target the virus Epstein-Barr virus, which is the kissing disease, causes mono, like many of us have had, but it also can target tumors because a certain percentage, 40 to 60%, I think, of EBV lymphomas are EBV positive. So these EBV T cells can treat viruses as well as virus-associated malignancies. But where I'm going with that is these EBV T cells, some elegant studies by Dr. Helen Heslop, Clea Rooney, and Kath Ballard back when she was at Baylor, they showed that these T cells persist in the body over 10 years. And so that's quite a long time. And these studies were done in the early 2000s or mid 90s when you could actually modify a T cell with a gene just for the purpose of tracking that T cell. Imagine, inserting a GFP protein into a T cell so that you could track it. Obviously the FDA doesn't allow us to do that sort of thing anymore, but these were done when it was still possible. And so they're able to look for that gene. It's not GFP. I think it's a neomycin resistant gene, but they're able to look for that gene over time in patients. And so as long as those patients are still living, we can try and detect those T cells that we know indubitably are derived from the T cells we infused and not from an endogenous T cell response.
To the best of your knowledge, who are the sort of oldest patients who've had a T cell therapy? In other words, how long has it been since the first patients were treated with this, where we actually can have data on kind of the longevity of a response?
Steve Rosenberg at the NCI, I think he was highlighted in a number of recent articles in the Washington Post and others. He's treated patients in the early 1980s. I think what's the more common T cell therapies really started in the early 1990s with Stan Riddell using cytomegalovirus specific T cells. That same trio I mentioned earlier, Clea Rooney, Helen Heslop, and Malcolm Brenner, using EBV specific T cells. And that again was in the early 1990s as well as Rick O'Reilly. And so I guess now they're 20 or 30 years out. And I think it's fair to say that a number of those patients are still living.
Well, I don't think I appreciated that it's such a rich history, but maybe I shouldn't be surprised because science typically does take a very long time to mature and come to fruition.
Yeah, but if you think about the history of these T cell therapies, it's only in the last, 7 years, industry has really been involved. I mean, there's always been, one or two startup companies, Dendrion, things like that have pursued vaccines mostly, but up until recently, it's been a number of these boutique cell therapy centers that were doing the majority of the research. And in order to get a cellular therapy, you had to go to NCI, you had to go to Baylor College of Medicine or St. Jude, you had to go to Fred Hutch, or you had to go to Sloan Kettering or UPenn. And that's just how it was, right? And so now with the advent of CAR T cells, everyone wants to be involved in cellular therapy. They see it as the next generation of therapies and this personalized medicine revolution.
It actually brings me to a point that topic that I discussed with another guest on our podcast, Aliyah Stupka, who's head of life sciences at Health Catalyst. And he's incredibly excited about this space too, but one of his kind of general concerns in the world is accessibility to treatment. And so you've talked about how these are now becoming more accessible, but as long as the price point is in the hundreds of thousands, there's still going to be kind of a limitation. How do you see the accessibility of biologics like this changing over time? What's your hope for people in, say, underdeveloped nations who have just as much need as the rest of us who have access to MD Anderson?
Well, I think right now we're all trying to fit these personalized biologics into a paradigm created by the FDA that was more suited for traditional pharmaceutical drugs, right? So tablets, chemotherapy, things like that. I have to give the FDA credit. I think that they're doing their best to keep up with rapidly changing fields. And they've come out with things like this RMAT designation and breakthrough and all these other pathways that can make drugs get to the market faster. And so that has helped cell therapies, I guess, get to the market. But to answer your question is, there are other paradigms that we could try that would make these drugs much cheaper, right? So we're still asking Novartis to go to 2 facilities or 3 facilities and make every personalized medicine, regardless of their location in the world, right? I think they'll probably have a couple more as they go into different countries, but it's a very centralized process. But what if that process isn't centralized? What if I were to develop a process at Children's National that is just an adjuvant to a bone marrow transplant, and it didn't need to go through full FDA BLA approval, right? What if the FDA were to come to my lab and say, you know what, you follow good manufacturing practices that are suitable for this type of development. We give you permission to give antiviral T cells or manufacture antiviral T cells and give them at your institution after a bone marrow transplant. And then we would just transfer that cost to the patient, right? So there's no middleman pharmaceutical company involved. And it would likely be a lot cheaper, because right now we're actually doing that at a cost to us so that we can treat patients who are in dire need of these antiviral drugs. To get to your point about accessibility beyond just these specialized centers, third world countries, developing countries, I think that's where we need to create the infrastructure, but we also need to create these off-the-shelf products. This is going to be a challenge. I don't know that I have all the answers and how to do it. All of our products are stored in liquid nitrogen. What kind of infrastructure do we need so that we can store these products in countries throughout Africa, throughout Asia, South America? I don't know, but I do think that a third-party product would be much easier to implement than a truly personalized one product per patient approach.
Yeah, no, that makes sense. I think for that kind of distribution, it needs to be commoditized at some point. I have a question. This is shifting gears a little bit, but I'm curious to get your take. So for some of us who follow cell therapy kind of passingly and in the news, the headlines, unfortunately, that stick, okay, they're the good news stories, but occasionally you'll hear about the kind of horror stories. And I think it may have been Juno or Kite who had a couple setbacks in some of their early stage trials. Now, I don't think this is that unusual. And frankly, I think these kinds of setbacks are often the sort of costs associated with progress. But how, as an industry, do you all kind of deal with the occasional sort of black eye in the press, but also the very real risks associated with, truly innovative treatments and therapy?
Yeah, I think, it is difficult to be innovative, but at the same time, provide a product that is going to be safe in every single patient 100% of the time. And that's a risk that we take. If you look at our patient population, the majority of the patients that we treat actually have already had a bone marrow transplant, which is very disruptive to the body, right? So without any additional therapy, a bone marrow transplant patient spends considerable amount of time in the hospital, sometimes in the ICU. And so if they're getting one of our therapies on top of that, we don't know if the reason they have an adverse event is because of the bone marrow transplant, which often gives them these adverse events anyway, or if it's because of a product that we gave, right? And so differentiating between those toxicities is in itself a challenge. And, is it fair to the patient that they don't get these potentially life-saving therapies because one patient out of 200 had an adverse event? I think that's something that we as a society need to appreciate. And some of the thoughts on addressing that are, so we have an institutional review board who looks at all the toxicities. We have to submit these toxicities to the FDA, so they're looking at it as well. And I know that IRBs often have community participants, but maybe that is an area where we have participants from the community come in, maybe from that disease population, and say, we understand the risks, and here's what we're willing to accept. We're willing to accept grade 1 graft-versus-host disease, maybe a little cytokine release syndrome in the case of the CARs, but what we really don't like is the neurotoxicity. And actually, the neurotoxicity is often reversible. That's the sort of thinking is, we can set up what we are not willing to accept. But like I said, a lot of those committees are already in place and they do review all the data.
Having worked at Baylor College of Medicine and myself and been around people associated with clinical trials, I actually have a lot of faith in the safeguards and the backstops put in place. And I think this is important for a general listenership to realize that some of these experimental trials may be experimental, but people are not generally being reckless. Perhaps one recent CRISPR study aside.
That's right. Let's not go there.
No,
But
leave that for another conversation.
I do want to point out too that for better or for worse, doing animal studies is often difficult, or is difficult in this situation, because when you have a small molecule, right? So you can have a murine model that has that cancer of interest, right? You can give the drug and see what happens, right? But when you have a biologic therapy, you need the human T cells to go into the mouse. And so in order to do that, you have to already have a very complicated mouse that doesn't have an immune system. And so then you can develop that cancer model in the mouse. But when you give our therapy, it's missing all the other cells that are naturally in the body, in the human body, right? So A, the T cells aren't going to work as well because they're missing a lot of the other immune cells that help orchestrate that response. And B, some of the toxicities that you might see, because there are other immune cells present, are not going to happen in that mouse because, again, they don't have the human cells to work in concert with the T cells you infused.
Certainly the case. And even with small molecules in mice, I mean, the joke, which is not even that funny, is that we've cured cancer in mice dozens of times. But there, at least you have kind of a systems biology that makes sense. And certainly as soon as you start thinking about immune modulation, it's a lot trickier. We see that with monoclonals. We see that with T cells and so forth. Let's shift gears a little bit more. So just as a disclosure to our listeners, Patrick and I have known each other for a while. I actually think we played on the same indoor soccer team sometime back in like 2011, 2012.
Yeah, when we were
both
young.
in Houston. Yeah, when I was never that fast, but I could get around a little better then. But so anyway, I follow Patrick on the social medias, and I feel like I've seen at least 2 trips of yours to Iran. Is that right?
Oh, yes. I just got back 2 Sundays ago.
Tell us about that. What are you going over there for, and what's kind of the scientific community like in that part of the world?
I feel very fortunate to have had the opportunity to go to Iran twice now. I think it's like any time you hear about people in the abstract, right? You develop a stereotype, and then when you actually meet the people, you hopefully say, oh, wow, this is not true at all. These are the kindest people I've ever met. And that's how I feel each time I go there. So not only are the people incredibly kind, but they're also doing really good science, and they're really hungry to learn about better ways to treat patients with cancer. They're really into using mesenchymal stromal cells in a safe and appropriate way with clinical trials. And, we're not talking about the stem cell tourism and things like that. Of course, there are others that I don't want to talk about today. I think every country has their own issues and crosses to bear. But, from my firsthand experience, I think it was a really great experience, and I couldn't speak any more highly of the people I met.
And what was the opportunity? Was it a conference or sort of a visiting rounds or something like that?
This last time it was the 3rd National Congress on Stem Cells and Regenerative Medicine. And then last year was a similar conference hosted by the Royan Institute. And actually, I was in Iran when Kymriah was approved, and I think it was August 30th, 2017.
That's exciting. It's for scientists like us, some of those dates are the kinds of things like, where were you during the moon landing? It's like, where were you when the first CAR T therapy was approved?
Yeah, I was telling Iran that it just happened, and it's going to cost $500,000 US, which is
good news and bad news.
Yeah. Which let's see, in terms of reals today, that would be something like 5 billion reals or something.
Have you gotten to do any other cool travel through your work?
Yeah, so I'm involved with a number of different organizations that allow me to travel. So one of them, I serve recently on the FACT board of directors. So FACT is the Foundation for the Accreditation of Cellular Therapy. And this is really interesting. Bone marrow transplant developed over the last, 60 years, independent of FDA review. So even today, bone marrow transplant is not under the direct purview of FDA. Because of that, you had a lot of different programs with varying degrees of quality. And so some of the pioneers in the field developed this set of standards for best practices, if you will. And they said, okay, let's try and bring everyone up to the same standard. And so you don't have to go to the MD Anderson's or Mayo Clinic of the world to get good care. What they did was they set up these standards, and they said, okay, we're going to hold everyone accountable to them. But by virtue of being an accreditation, it's a peer review process. We are FACT accredited for minimal and more than minimal manipulation in our cell therapy lab. We have immune effector cell accreditation for our hospital, and just overall FACT accreditation for our bone marrow transplant immune cell therapy program. But what that means is, since I'm accredited, I can be an inspector. And so I get to go to other programs and inspect other programs. And I will say that I learn a lot from these inspections. So I probably learn more from them than they learn from me when I'm doing the inspection. But that's the beauty of it. You get to network with people. You get to see, how does this group handle training? Because it's a big challenge for all of us. How do they do their audits? Do they go on site to every vendor and do a formal audit, or do they submit a questionnaire, ask if they've had any 483s from the FDA and call it a day. So a lot of my travel, or 2 travel trips a year, is because of the fact I get to go and do an inspection throughout the world.
No, that's fascinating, and probably a great way to spread your expertise and actually help others do this important work.
Yeah, as we try and get more people to go into cell therapy, because there's certainly a shortage of qualified people, that is absolutely one of the perks, is you get to travel the world for free and meet a lot of really cool people.
Let me ask you to sort of put your Mana hat back on for a second. I'm interested to just hear a little about your experience so far with venturing into the world of startup. Having come from academia and then I guess academic clinical center sort of role. How are you finding it? How much time do you actually have to dedicate to it?
Yeah, so it's exciting, right? So finding time to dedicate to it is not a problem because you want to do it. It's something I have trouble explaining, but once you step into it, you're like, wow, this is fantastic. It's like anything you're passionate about, right? You're passionate about it, so it isn't a problem. You look forward to doing it. It's not like me trying to go for a run in the morning. Not only can I not wake up in the morning, but I also don't want to go for a run. So finding time is not a problem. Sometimes if I have to travel, then it becomes more of a problem, taking calls. So I tried to take a call from Iran, but I couldn't figure out how to use my telephone. So wasn't able to take that call at 2:30 in the morning. But otherwise, it's just about time management. Typically I'll spend whatever, come into the office between 8 and 9 and leave, 6, 7. And then I'll go home and answer all the Mana emails that have been accumulating throughout the day. But, it's been such an exciting journey that I don't mind it. It's also forced us to really analyze all the data that we've accumulated over the last 4 or 5 years, which has been helpful as well.
Yeah, and that was actually going to be one of my sort of next questions. And this is something I ask of a lot of people who either continue to straddle 2 gigs where they have an academic position and also an entrepreneurial one. What do you find that the startup allows you to do that, say, is just kind of prohibitively bureaucratic or just not even possible in the academic life?
Ooh, all right. Move quickly. Throw money at things because we want it to happen tomorrow. I think that's the biggest thing. It's not just our startup company. I asked someone once how many people they had on their process development team. I think this was one of the CAR T-cell companies. And they said they had 60 people just on process development. And I said, that is crazy because we have, 15 clinical trials at Children's, and we probably have 0.5 people dedicated to each trial in terms of the manufacturing. So, that's the difference. If you want to get something done in, given your budget and your timeline, then you just throw money and people at it and you solve that problem very efficiently. I'm happy where I am because I get to be involved in both worlds. I'm not sure that I would be able to just focus on one small part of a big problem. I like being involved in all the different parts.
I definitely also enjoy the many hats aspect of it. But that's a really interesting point. And, back in the day when I used to cook for a living, I would kind of joke the difference between cooking school and just going to the restaurant is in the former you get an onion to chop, and the latter you get a box of onions to chop. So you get faster doing one than the other.
That's true.
No, that's fascinating. I'm excited to see, when you guys emerge from official stealth, it looks like you're off to a great start already. Definitely have a founding team to be envious of.
Yeah, I'm really happy. I think, we wouldn't have done this if we didn't have the right people and if we didn't want to treat patients. I think everybody wants to make money and things like that. But I think the biggest driver for us was there is a line of patients out the door that need to be treated at Children's, with our innovative therapies. And we just don't have the space and the people and the time to treat all these patients. And so we said, we should start a company, find a different manufacturing facility, and be able to treat these patients with the therapy that we think is going to be transformative.
Did you license out the technology from Children's or from Baylor or elsewhere?
Yeah, so it's the license, the IP is held at Children's, and then Mana licensed the IP from Children's.
Gotcha. I don't need you to speak ill of your current employer, but is that generally a model that Children's is comfortable with? Or do they have a history of kind of supporting innovation and tech transfer and so forth?
Yeah, actually. So they, a few years, 10 years ago, something like that, they received a $150 million gift from the Sheikh Zayed of the Emirates. And with that money, one of the things that they did was they set up this Sheikh Zayed Institute for Pediatric Surgical Innovation. And the focus of that institute was actually to drive innovation and have spin-out companies for ways to do precision medicine that didn't involve surgery and things like that. And so they've really encouraged people to have startup companies, to have IP. Because of that, they've set up this nice little incubator type program.
That's really fabulous. And I'll just get on my soapbox and say, for anyone who is listening from academic administration, if you want to attract the best young talent to teach and do research at your institution, support innovation. So Patrick, one last question. How do you see the kind of assist from the movement in big data and public data commons and sort of applied mathematics and machine learning helping cell therapy?
Yeah, so that's a great question. I was recently in London for this Pediatric Innovations in Healthcare meeting, and Atul Boot from UCSF gave this lovely presentation where, since he's in the California health system, he's able to take all big data and basically analyze it, right? And say, okay, throughout the entire California healthcare system, these patients all got this frontline treatment for diabetes. And then how many patients then went on to the second treatment, and then a third treatment, and then a fourth treatment. And then using that big data, he can say, okay, well, clearly this person that got this as a second therapy went on to 7 subsequent treatments. So why the hell are we even giving that second treatment to begin with if it's not working for anyone, right? And so that was mind-blowing for me. So we can do that same information using our cellular therapies, right? So we can look at, okay, so this patient gets a bone marrow transplant, right? And here are all the patients who are going to relapse. Now, let's give them our T-cell therapy. And let's just say we target one protein. Does that work? What if we try 2 proteins? What if we try 3 proteins? And so using that big data and a lot of patients treated, we can figure out what the best way to approach is, rather than just sort of trying to guess whether it's working or not. And one of the questions you asked earlier was persistence of these T cells. Well, one of the questions we're still trying to address in every therapy we do is one infusion enough? Or if the patient responds, should we give a sort of preemptive second, third, fourth, fifth dose? And I think this is a way to answer that, is using the big data, see if people go on to relapse. And in some cases, you don't even need to go and do a follow-up with the patient, because it's in the medical record system. And as long as you have consent to access that data, you know the answer.
Yeah, no, that's fabulous. And I think that the more and more we can use real world data and patient outcomes to inform both the development of and delivery of these therapeutics, the better we'll be. Patrick, this has been fantastically interesting. I think you guys are doing great work over there, and I appreciate you taking some time to talk with us today.
Yeah, no, it's been a pleasure to speak with you again.
This has been episode 6 of Talking Precision Medicine. Please share it with your colleagues, leave a comment or review, and stay tuned for the next one. Thanks for joining the conversation.